was going to talk about that.
Yes, I totally forgot. Yes, okay, I'll give you a minute then.
Okay.
Is this an honors project? Or
yeah, I guess.
This semester, or?
I asked for this semester. I technically already did the honors project, but we're just still working on it. Hopefully we'll publish something in a journal, so that's the goal now.
Okay.
Yeah. So it's kind of funny because I'm not do BMS, but I'm at BMS. I had your class for microbiology last semester.
Okay. Yeah, I recognize you. Okay. Okay. Yeah, whenever you're ready. So go ahead and. So good afternoon. We're going to have Gordon is here just a couple minutes of your time before we get started to talk about a project that he's working on. So.
Yes. So, hello, my name is Gordon. I'm a BMS major. I took this class last semester, so how fun it is. Yeah. Anyway, I'm doing a project with a professor, Dr. Corber. We're just interviewing students about disability support resources, DSR. So if any of you use DSR and would like to share your experiences, we're conducting interviews with students and we're hoping to publish the results. So if you have questions or would like to participate, I got these sheets with the QR code. I'm not sure if I should send them around or.
How about, I don't know, you want to put them right up here?
I'll put one just in the back. Yeah, that's all.
Okay, great. So yeah, if you have had experience with DSR, Gordon's interested in hearing about that. So to help him out. All right, thanks for coming by.
Thank you.
All right, so where we left off last time, we had finished up our gram-negative cocci. We finished up the gonorrhea, Neisseria gonorrhea, and the Neisseria meningitidis. And we started the Bordetella pertussis, which is another gram-negative pathogen. Remember, though, it is a coccobacillus. So partway between a round cell and a rod-shaped cell. Humans can be carriers. We talked about the way that infection progresses. We have specific names for the prodromal period. We call that the catarrhal stage. Catarrhal means infection or inflammation of mucous membranes. That's when a patient with pertussis is the most contagious. And that's just cold-like symptoms. So that contributes to it being passed. People don't really know they're very sick yet. In that paroxysmal stage, I hope we took a chance to listen to that whooping sound. Young children and babies get whooping cough. A paroxysm is a violent attack. So that's the illness stage. That's when they're the most sick. Even when adults get this, remember, they're not as ill. They call it the 100-day cough. So they're ill for a long time, but not as acutely ill, perhaps. So big difference in babies and adults. Transmitted, not airborne. A lot of these respiratory infections, you may think they would be, and they have the potential to be, but we think that's not the most common way. Okay, tuberculosis, yes, airborne. But at least the Neisseria meningitidis and as well as this Bordetella pertussis, we got to be typically pretty close contact, within a meter. It's the big droplets, the coughing and the sneezing. We seem to have kind of every once in a while have a peak year of Bordetella pertussis. So the last peak year was in 2012, where we had about 48,000 cases in the US, only 20 deaths. So again, it's usually really young children that are more susceptible to dying to this. In 2019, we had, so last year that I have numbers for, about 18,000 cases. So a little bit less there and only 10 deaths. So pretty rare. Before we had a vaccine in the US, our first vaccine came out in like 1940. Before then, we had something like 200,000 just kids get sick in the US and closer to like 9,000 deaths. So that was before vaccine, before antibiotics as well. And of course, worldwide, the cases are in the hundreds of thousands. So this is still an issue worldwide. We're seeing these spikiness, a little bit of increase from year to year because we're using a different vaccine now. And we'll talk about that in a second, but that protection wanes a little bit. So we tend to have a little bit higher incidence, even though maybe we're getting more people vaccinated. We didn't talk about virulence factors yet. So what makes this organism successful? There's quite a lot of them, and I picked out a few. I think this organism produces something like 8 or 9 different exotoxins. So it's a really important part of its virulence. Pertussis toxin is the one that's easiest to remember. Okay, so, but they all of these toxins that the organism secretes, they have some similar function. Okay, so all of these either are mostly working to help the organism adhere to host cells or dampen innate immunity. And you know that when our innate immunity is dampened, then that's the way that our adaptive immunity gets activated. So we don't have the innate, then we don't get as good adaptive immunity, as good antibody production. So things like helping the organism bind to epithelial cells. We see the organism here in this picture, the kind of light greenish, they kind of look a little bit more like rods in that scanning electron micrograph there. But it's really important that the organism binds to its target cells. So it has a lot of different mechanisms to make it successful to do that, because it doesn't typically invade host cells. It stays bound to the outside of them. It has some, blocks the migration of white blood cells to that spot by these exotoxins, and inhibits killing by our phagocytic cells like macrophages, neutrophils. Interestingly, increases mucus production. So we talked about how that mucus is important to help trap microbes, and at least in the respiratory tract, then they get kind of coughed out and expelled. Well, it turns out too much mucus makes it a lot harder for us to cough them out. So too much mucus for us is a bad thing. It also inhibits that cilia movement. So again, we have a hard time expelling those pathogens out of the respiratory tract. Okay. Another, it's a protein. We call it an adhesin, filamentous hemagglutinin. And I want to mention this one because we actually include that protein, or at least part of it, in the vaccine, along with an activated pertussis toxin. This is actually located in the membrane. It's associated with the outer membrane of the cell wall. So we call it a protein. Is it really an extracellular enzyme? They don't really call it that, even though sometimes it does get secreted a little bit, but it's a structural protein in the outer membrane of the cell wall. And that's another main function, helping the organism stick to those ciliated cells. It has some inhibitory function to neutrophils as well. So, and that, if an organism if the Bordetella cannot make that protein, if they made mutants that can't make that protein, then they can't stick to the epithelial cells in the trachea. Okay, so they're not able to colonize the trachea if they don't have that protein. So really important protein. And then like the Neisseria, because they're gram-negative, they have endotoxin, that slightly different structure. Instead of lipopolysaccharide, they call it lipooligosaccharide, just a little shorter sugar subunits there. And again, that's responsible for inflammation. It's that lipid A that causes inflammation. And I was thinking a couple of days ago, why does that help the organism to increase inflammation in our bodies? You think they would want to maybe dampen the inflammation because it's through inflammation that we get the white blood cells where they need to be. I think it's just kind of a side effect. That endotoxin, that lipid A, is part of what makes that outer membrane of the gram negatives so impermeable to a lot of things. Okay, so it does have function as structurally to that organism, and kind of a protective function, and it increases inflammation, which sometimes too much inflammation helps the organism. So it's kind of a side effect there. All right, how do we treat this? Do we have a vaccine? It seems like there are several different antibiotics that can be used, so I won't worry about naming which antibiotic. But we don't have a lot of resistance to Bordetella pertussis, so that's a good thing. We probably have a variety of things that can be used. Interestingly enough, that the antibiotics don't seem to increase the time it takes for a patient to get better. It takes usually takes about 2 weeks to actually kill the organism. But we talked about how the organism produces so many exotoxins. Does the antibiotic act against exotoxins? No, right? So it takes a long time for those exotoxins to kind of get out of circulation. So it really, I didn't give myself enough space to room here. Right, so it does enough room to write. That's what I was trying to say. So it does not deactivate the toxins, those exotoxins, and really the antibiotic does not shorten recovery time. So why do we give patients antibiotics? Because it lessens the chances that a patient will pass the infection on to someone else. Say will be contagious. Okay, they're less contagious, that's what I'm trying to say, if they get on antibiotics. Okay, and typically they'll give, if somebody was exposed to somebody with whooping cough, typically they're given prophylactic antibiotics just to try to head that off. All right, so best to prevent this infection, this original vaccine that we have always put together with tetanus and diphtheria toxin in the form of DTP, that was the form that came out in the 40s. That, like I said, included the diphtheria and the tetanus toxoid vaccine. The pertussis, it was a whole cell killed vaccine. So we talked about that, really quite effective. The problem was because of that endotoxin that was associated with that whole cell, that there was a lot of inflammation, not serious inflammation, but enough that when it makes a baby cranky, people don't want to get it. So they switched to this DTaP, that was in the 1990s. That AP stands for the acellular pertussis. We consider that a subunit vaccine, even though it has inactivated toxin in it. You could consider it at least partially a toxoid vaccine, but you always hear it referred to as a subunit vaccine because it's also got part of that filamentous hemagglutinin in it as well, that outer membrane protein. Okay, so we have to give more doses because it's a subunit vaccine, typically a series of 4 different shots. Start that pretty early, try to get babies some protection. Unfortunately, the immunity, the problem is the immunity wanes by the time those babies are maybe 10 to 12 years old. Okay, so essentially it protects them for a while. So it's recommended that when kids get to be about 12 years old, that they get a booster. And that is often given in a little different form. Sometimes all the T's and the D's and the A's and the P's gets a little bit like, well, wait, which is which? But this is the Tdap, that's a little bit of a lower dose, apparently, for, again, a booster for young kids. It's also recommended that any adults that are going to be taking care of a baby or pregnant women in their, let's see, between 27 and 36 weeks of pregnancy, get the vaccination as well. And that way, at least, not only will the mother be immune, but she can pass some antibodies through the placenta and protect the baby in the early months of life. So what kind of antibody is that passes through placenta? IgG, right? That's the one, that's the only one that passes through. Okay, so good. Any other questions about pertussis? Okay. All right, let's pause before we get into our viral pathogens and do a couple of review questions. Okay, so take a read there. Take an answer. I think it's a good time to mention, especially as those of you are thinking about your lab exam coming up, remember our grammar when we're writing organisms' names, okay? Italicize when you're typing genus and species, uppercase genus, lowercase species. Your handwriting, you just underline, okay? And interesting, when we say something like gonococci or streptococci, that's kind of a general term for any species in that genus, and we don't capitalize or italicize that or underline. It's kind of a lot of rules to remember. Okay, so which of the following statements is false? True, it causes disease in humans only. That's true. Fimbriae, a really important virulence factor. That's true. They secrete protease that destroys that antibody that's most prevalent on the mucous membranes. That's true. The one that's false, lasting immunity. No, you can get it over and over again. And that's why we don't have a good vaccine for gonorrhea. Okay, and this is true as well. Infected women are often asymptomatic. Men can be as well, but more often women. Okay, I can get a lot of information in one multiple choice question. Okay, here's one, read that over, Neisseria meningitidis. This one's a little trickier, maybe. What do you think? Anybody willing to share what they think? I think that's the tricky one. That's not a bad answer, but it's not the best one. What do you think? Yes, the answer is D. So remember, we talked about this organism can secrete endotoxin kind of in a weird way that other gram-negatives don't. What do we call that process? It has a special term for it when it's releasing endotoxin. Okay. Called that blebbing. And that endotoxin is 100 to 1,000 times more toxic than kind of run-of-the-mill endotoxin for gram-negatives. Okay. So it does invade the central nervous system. That's true. But what about that? Oh, it's because it's releasing the endotoxin. So, okay. So again, sometimes you got to pick the best answer. Okay. This one should be easier. Again, review a few terms here. Oh, look at that. I did not italicize. Oh, I'm so embarrassed. Just talked about it. I think I spelled it right, which is an accomplishment. What was correct answer here? D, yes. So we called it an eye infection, right? Ophthalmia neonatorum. That's the correct answer. A bubo, that we didn't do bubonic plague this semester, so you shouldn't have recognized that. An eschar, remember what an eschar is? That's like a black scab. That's as a result of cutaneous anthrax. Okay, and petechiae. What is that symptom? Sign of? That's Neisseria meningitidis that can cause the kind of a black rash because of the broken blood vessels, broken capillaries. Okay, so again, those terms, one more. Okay. A little bit, again, more straightforward, just testing your recall. This one you might guess just from the name of it. Yes, C. So that stands for meningococcal ACWY. Remember, those were the different serotypes of the capsule that are the most common, along with B. All right, Prevnar 20, that was the one. Just saw a commercial for that last night. Again, Streptococcus pneumoniae. And then BCG, that is a vaccine against tuberculosis. That's the TB. Yeah, question. So you asked a question about MenB. Yeah, so that we do have a separate vaccine for the serotype B, but for some reason they weren't able to bundle it in with all the rest of them. So it's a separate shot. Yeah. And then we just talked about this, that's tetanus, acellular pertussis, and diphtheria. So again, the BCG, that's the TB. Okay, so good, keep reviewing. That's okay. Any questions there before we move on? All right, so let's start some viral pathogens. I have kind of lined up only five different more pathogens to talk about. If we finish a little bit early next week, we'll do some more reviewing, but I think that seems pretty reasonable. So just sometimes we get caught up in details and I think forget kind of big picture, what's really important to take away from this class. So reviewing some of those big picture things about viruses before we talk about our first DNA virus. Remember, viruses, they can only reproduce inside a living cell. All right, so we can't grow viruses in the lab just on, say, nutrient broth or something like that. They have to be within a living cell. It can be a bacterial cell or some other kind of cell. What's the structure of a virion? Remember, the virion is when it's outside of a cell, kind of all together, and then we would correctly call it a virus when kind of the outside parts of the virion come away from it. They're going to be, a virion is going to be composed of nucleic acid. So either DNA or RNA. No viruses are going to, no virions are going to have both of those things. They're either one or the other. It can be double-stranded or single-stranded, either one. And what do we call that proteinaceous shell that's around the nucleic acid protecting it? Starts with a C. Capsid. Okay, so a capsid surrounding that protein. Those little capsomeres are proteins that make up the capsid. Okay, several different morphologies of capsids. They can be helical, they can be polyhedral, like this one here, most often icosahedral, meaning 20 sides. Okay, that's a very stable structure. Or complex. There's a few that just don't really fit into either one of those categories. Let's see, what else? And then sometimes virions can have an envelope, can have a phospholipid membrane around them. Usually bacteria that infect, or sorry, viruses that infect bacteria, bacteriophages don't have an envelope. Very often viruses that infect animals do. Not all of them. Things like norovirus doesn't have an envelope around it, but more often they do. Okay. All right, and then this was just, you know, there's a ton of different viruses. We're going to start out and we're going to talk about 2 different DNA viruses and then several different RNA viruses. There are, in general, more RNA viruses. But Herpesviridae, so that is a family of viruses. Remember, when it ends in this viridae, that's describing a family, species, genus, family. That's the next taxa up there. It's kind of awkward trying to put viruses into categories the same way that we do with living organisms, but we try. So this is a huge family of viruses. We're gonna focus in on several different ones. You'll find a few questions in your homework in the chapter about viral infections of the urogenital tract. But they talk about it in your book in several different spots because it infects different kinds of tissues. So, all right, so what can we say about this organism? First of all, sometimes we hear this referred to as herpes simplex virus. Our old book talked about how they have changed that nomenclature. Now we refer to it as human herpes virus, but apparently both names are still in use. You might hear human herpes virus, or you might hear herpes simplex virus. Apparently both names are used, so that's always a little bit confusing. What can we say? What are the notable characteristics about this organism? It is enveloped. It actually takes its envelope from the nuclear membrane of the host cell, which is a little bit unusual. Most oftentimes it's the plasma membrane. It has an icosahedral capsid. Okay, so you can see that in the diagram there. And it has linear double-stranded DNA. Okay, so what I mean by linear, you can see here in this graphic, the DNA is all in one strand. Some other viruses will have different segments of their nucleic acid. Okay, and this is one of the larger virions, and it also has one of the biggest genomes. So somewhere between 80 and 100 genes. Okay, the smallest genome of any virus only has 4 genes. So quite a bit. It comes prepared, packing lots of its own genes. Okay, another interesting thing about the Herpesviridae, they all have a very prominent tegument. That's not something that we've talked about before. Apparently, I think there are other virions that have a tegument, but it's just especially prominent for Herpesvirus, Herpesviridae. And that's just a proteinaceous region outside of the capsid, yet inside of the membrane. So in this diagram, that's all those little purple circles there. Okay, so they carry in with them some proteins when they infect the cell. And these are proteins that are especially important to suppress the host immunity and also aid in viral replication. So I tried to look up, well, what exactly do some of these proteins do? Some of them will suppress something that we've talked about, type 1 interferon production by the host. So remember, type 1 interferon, alpha and beta interferon, those are cytokines that are secreted by an infected cell that will tell cells around them, hey, kind of prepare yourself, and they make this inactive protein that becomes activated when they get infected with a virus and then causes them to stop their protein translation for a time. So it tends to really kind of slow down the spread of viral replication. So here's a sneaky thing that herpesvirus does is tamp down on that cytokine. Another thing that some of those proteins do, that I referred to this in one of the practice questions that I gave you for the last exam, it actually, some proteins act to down-regulate MHC class 1 expression in the host, say in host cells. Okay, so again, sneaky, that's the way that an infected cell would advertise that it is infected. That's the way that a cytotoxic T cell would know to target that cell and kill it. Okay, so again, that's a way that herpes virus helps it not be detected by our immune system. And then, like many virions, there's some glycoprotein spikes. We won't talk about them real specifically like we'll get to for influenza, but just, you know, recognition of host cells, certain kinds of host cells help the virion bind to it and enter into those host cells. What else in general about herpes viridae? This is a family of virus that's very widespread. One source I saw estimated that close to 100% of us have been infected with some herpes virus. Sometimes we have subclinical infections with no symptoms, and so you may not have known it. And it doesn't always go latent. Sometimes we may fight it off before it has a chance to go latent. But it is quite widespread. The reservoir, humans. Okay, so I'll say except in rare instances. Seems like there's always exceptions, right? So for the most part, humans are passing herpes viruses to other humans. We are not getting them from animals. They have found there is one variety of herpes virus that I was reading about in macaque monkeys. That in macaque monkeys, they get, like, you know, fever blisters or cold sores, but when humans come in contact with them, they get this nasty encephalitis that is often deadly. So there has been about 20 people that have had this kind of weird transmission from monkeys, but very rare. Okay, so mostly humans give it to humans. Here's another cool thing, or maybe not a cool thing, I guess, in terms of our health, but all herpes viridae will show latency. So remember, latency in animal cells or animal viruses that infect animals, that's what we call lysogeny in viruses that infect bacteria. So remember, in that lysogenic cycle, the viruses can integrate into the host chromosome and then just kind of hang out there indefinitely every time the cell divides. And then eventually in bacteriophage, they can come out. They can, of that lysogenic cycle, and actually lyse the cell eventually. But for viruses that exhibit latency, they're never induced. They never have induction. They don't ever come out, as far as we know. Okay, now herpes virus is a little bit different the way it acts, though. It doesn't actually incorporate into the chromosome. It forms these little episomes, which are plasmid, kind of plasmid-like structures. So plasmid-like circular DNA. So if here's a host cell, let's see, here's the nucleus of maybe one of our cells, and we've got chromosomes, okay, it will form these little circles. Okay, that's what that's supposed to be right there, that little circle, that's an episome. So the episomes are in close proximity to our chromosomes, but they're not actually integrated into them. But it has the same effect. Once they're there as episomes, they don't ever come out. Okay, so in other words, this is permanent. I mean, who knows? Maybe someday we'll figure out a way to get rid of it. But for now, so the best we can do really is to manage our symptoms once we're infected. Some of the herpes viridae are oncogenic. There's actually 8 different herpes viruses. We're only going to talk about 4 of them. One of them that we're not really going to mention anymore causes a Pertussis sarcoma, which you hear about really only AIDS patients having that, but that's caused by HHV8 actually. And then one that we will mention causes a cancer of the jaw called Burkitt lymphoma. So we'll talk about that a little bit more soon, but some of them actually cause cancer. This is a nice little 2 or 3 minute animation that is very well done. The sound is just kind of cumbersome in here, so watch that sometime on your own, but it really helps you to understand kind of what's happening when the virus, how it enters the host cell, kind of shows it, you know, making its proteins, and then how it leaves the host cell. So it's very well done. So if you're interested. Before we get into the specific infections, a few more characteristics. So they target mucosal membranes. So any little tiny microscopic tear in one of our mucous membranes will allow the virus access. How often do we have, especially in the winter, maybe little cuts in our lips or our cuticles of our fingers? Okay, so all the virions target the mucosal membranes. They multiply in the basal and epithelial cells in the region. So epithelial cells, they're just the type of cells that line all the surfaces of our body. So, you know, they're, you know, the skin, blood vessels around organs and mucous membranes, for example. And then basal cells, that's the bottom layer of the epidermis. So they're the cells that make skin cells, essentially. So we have the virion multiplying that causes inflammation in the host and lots of cell death and further infection of more cells. How we diagnose, your OpenStax goes into some detail about how we look for herpes virions with electron microscopes. And my understanding is that that's a very cumbersome thing to do. Maybe they have easier ways of doing that now, but in the past, we've usually looked for some kind of cytopathic effect. Sometimes we abbreviate that CPE. And there's lots of different cytopathic effects, but that just means a virus-induced cellular change. That's visible using light microscopy. Okay, we look for there's different kinds of cytopathic effects. For instance, rabies virus causes these little aggregates of proteins called megribodies to be visible when we look at cells under light microscopy. But the one that herpes virus causes is called syncytia. And that just means multinucleated cells. So you can see that in both these pictures here, all these little dark purple circles there, those are nuclei. So that's a cell that's growing in tissue culture. I think that's why it's kind of spread out like that. But you can see lots and lots of nuclei there. Here, this is, I think, some kind of tissue sample from a patient. You can see all these nuclei. And why do we see multinucleated cells with this infection? If you think about it, this is a really crafty way of the virus to avoid our immune system. When the virions are outside of the cell, that's when they are most susceptible to maybe being phagocytized or having antibodies stick to them. If they can stay inside of the cell, they're more protected. So this is one of their mechanisms. An infected cell will actually fuse with another cell that's not infected. So those virions never have to burst outside of the cell. They can just kind of stay intracellular. Okay, so they avoid detection by the immune system that way. Okay, and again, it's much easier looking at samples under light microscopy and just instead of looking for the virion itself, you're looking for changes that it's made in our cells. All right, so if we look at the different viruses, there's a number of them and they cause very different kinds of infections. So like I said, there's 8, sometimes we count 9 different human herpes virus, and they kind of go by different names. Again, sometimes we call them HHV, sometimes we call them HSV. But HHV1, that is the causative agent of cold sores. And then HHV2, that genus is the causative agent of genital sores. Now, occasionally you might have cold sores caused by HHV2. Typically, that is only when the patient has engaged in oral sex. So it's not a hard and fast rule, but generally what we see, HHV1, cold sores, HHV2, genital sores, above the waist herpes, below the waist herpes, you could think of it that way. HHV3 is also called varicella-zoster virus. That is also known as, anybody recognize that one? Chicken pox. I heard it from somebody. Yes. People of my generation are kind of maybe more interested in that than people of your generation, because most of you probably haven't had it. And then HHV4, Epstein-Barr virus. Some of you have had that. That is the cause of infectious mononucleosis, among other things. So we'll talk about a couple different things that virus can cause, kind of briefly. All right, so let's talk a little bit more about HHV1. Okay, how do people get it? Estimated 65% of the US population has been infected. Many are asymptomatic, but this causes, the kind of official name is gingivostomatitis, but we would call that cold sores or maybe fever blisters. Okay, they break out on the lips or maybe the skin around the lips, sometimes just inside of the mouth. Kind of painful vesicles, apparently. They are transmitted by close contact. So kissing, for example, or maybe some fomite. Remember, a fomite is an inanimate object, something like a drinking glass or sharing eating utensils, forks and knives. Okay. Usually the sores last maybe 7 to 10 days. Oftentimes you might have a little prodromal period where you feel a little tingling and you think, oh no, I think I'm getting a cold sore. A little itching or tingling. HHV1 can manifest in other sites other than the lips. I think when we get it on our fingers, we call that Whitlow disease. Let's say that enters through the cuticle region generally, and that's the primary region that dental hygienists wear gloves. And really lots of reasons why they might do that, but medical professionals too, to try to keep from getting herpes from patients. That can easily be transmitted to the eyes. How often do people rub their eyes? The disease there we refer to officially as herpetic keratosis or keratitis. So you can see that lesion there. That can lead to people being real sensitive to light, and can also lead to permanent vision damage. So that's definitely something to see the doctor about. In really, really rare cases, HHV1 can infect the brain or the spine. We call that herpes simplex encephalitis. Most often, only somebody that's really immunodeficient would be susceptible to that, but that does cause substantial mortality. Okay, so again, quite rare. I spared you the graphic pictures here, which HHV2, but that's the causation of genital herpes. Okay, so lesions on the genitalia. How common is that? The numbers that I have come up with in the US, 1 out of 6 adults. Okay, so perhaps not quite as common as chlamydia and gonorrhea, but still quite common. So targets the mucosal membranes of the urogenital tract. People would typically have 2 to 3 outbreaks a year. The first time they have an outbreak, that often will typically cause a flu-like symptoms along with the lesions. And then when it reactivates, maybe 2 or 3 times a year. Although some people have reactivations as often as 2 to 3 weeks. So there's a lot of variability there. And it can be spread to sexual partners even when there's no active lesions. Okay, so again, let me just transmitted by sexual intercourse, in case that's not obvious. Okay, so it's more likely to be passed when there are lesions present, but it potentially can be even when there's no lesions. Itching symptoms there, itching, discharge, sometimes fever. Babies can get neonatal herpes during the birthing process. They can get an eye infection that kind of looks like gonorrhea in newborns, but of course the antibiotic drops aren't going to do any good if it's herpes. And it has the potential in babies to move to the central nervous system and can be life-threatening. So for those reasons, if the pregnant mother is having an outbreak at the time of birth, obviously they'll take the baby by cesarean section rather than birthing them vaginally. A mother with genital herpes statistically has a higher chance of having a miscarriage or delivering preterm. So. Okay, so just to wrap up here today, all the herpes go latent. They will go latent in nerves. So for HHV1, typically they're going to go latent in one of the 3 different branches of that cranial nerve number 5, the trigeminal nerve, either the ophthalmic branch or one of the jaw branches. Okay, and then, you know, whenever there's some stress, the virus will start to replicate again, and you'll have another sore. Whitlow will often go dormant or latent in the brachial ganglia, and then HHV2. More often, Whitlow is HHV1, but it can be either one. And then HHV2 will go latent in the sacral ganglia. Okay, so we'll talk about the sorts of things that can cause reactivation there. We'll move on, finish the herpes next time, and get into smallpox, I hope. So have a good weekend.